So hello and welcome back to A Tale of Genetic Code. And we'll be making a new video today on microscopes. So here we are actually switching tracks. We are stopping videos on molecular biology from Clark for a while so that we can cover cell biology in more detail before we can go back to molecular biology and understand it in a better manner. So for that, the very first chapter in Dalton and Young is on microscopy, and it is quite interesting. And let's just get into it, right? So what is microscopy? It is essentially the techniques that we use to see really, really small things. And why would we be interested in living in looking at very small things? Because those things that life is made up of is very tiny, and those things that affect life in a very profound manner, like bacteria, viruses, and even disease states, which we don't really understand well, but might affect a cell in a manner that is recognizable by just the look of it, and they cannot be seen by the naked eye because the wavelength of light that our eyes are capable of seeing, they are not basically sufficient to look at such small details in things. So we require instruments basically to look at really small things. And this slide just gives us the range of sizes that are available in the universe. At one end, we have the universe itself, which is estimated to be about 10 to the 27 meters large, which is really an astronomical number, pun intended. And on the other side, we have the Planck length, which is extremely, extremely microscopic. Again, pun intended. And somewhere in between, we have the really interesting parts, interesting because they affect us in a very palpable way, and they are also very easy to study, like the things that we are interested in, right? So we might be interested in studying the ribosome or the DNA or bacteria or our own cells, and all of this is possible to be studied because of instruments that we have devised, which uh, take into account the size, which we take into account the size, and then decide which of these instruments we are going to use. And we'll see how these instruments are actually working and how they are selected for these purposes. So, what is microscopy? Microscopy is essentially the field where microscopes are used to view objects that are too small for seeing with our own eyes. And there are a host of different types of microscopes, and we'll be seeing some of the most commonly used ones in this video and understand how they work and what they can tell us about the things we are interested in. So before we go on to actually see the different kinds of microscopes, we need to understand some few concepts that are critical to understand the functioning of the microscope. So the first is magnification, and magnification just tells us at what the actual size of the object is and how big it appears when we use this instrument to look at that object. And if you divide the apparent size by its actual size, you actually get the magnification that that instrument is able to give you for observing a, a particle of interest, right? And the second important concept is resolution. So uh, you can imagine two pictures, like you can see them here itself, that appear to be same, but you can say that there is some difference here, right? This image appears more clear and of a better resolution than this one. And that exactly is what resolution is. It is the ability to distinguish two closely spaced points or lines in an image. So what that means is that there is a higher density of information in that image. And that is why it has more resolution. So in photos taken up by your mobile phones, you would measure the resolution in pixels or as a proxy of that in the size of the image in the units of megabytes or kilobytes, right? And these two things are actually kind of antithetical to each other. So when you try to increase magnification, the resolution tends to come down, and or the vice versa. So when you're deciding upon which uh, which microscope or which instrument you have to use to conduct the experiment that you're interested in, you'll have to take into account its magnification and resolution powers before you can actually make the right judgment in choosing one of these. And now there is a concept of the particle. So we are using an instrument, and for example, your eye is an instrument, and you use your eye every day to look at things around you. And the particle that our eye uses to make sense of the world is called the photon. So basically, there is a source of light somewhere around you that is emitting photons. These photons are bouncing around in your room in straight lines, and from the last object they fell on and bounced off of, they are reaching your eye, falling on your retina, and your brain is making sense of it as an object that is around you. So this is essentially what a microscope also does, except that it just increases its size because of the various lensing mechanisms that microscopes have. So when you use photons like your eye does to resolve and magnify objects that are too small for your eye to see, that is called light microscopy. And instead of photons, when you use electrons, that is called electron microscopy. It's as simple as that. You might ask, why would you use something like an electron? Isn't electron like in the wires and like you get shocked when you touch electricity, and it does not just seem very intuitively like something you would see with, and that is actually correct. But if you think about it, you can make sense of the world around you in any of the several mechanisms that uh, it might be possible to use to do this. So if you have watched the series called Limitless, which is actually a sequel to the Limitless movie, which is really good. I think everybody should watch it. You see a man who had uh, basically reconstructed a person's face because he just remembered touching the face of that person after taking that NZT pill. And so that what that tells us is that uh, shape and what things look like are just information which can be got by many different mechanisms. And when you can throw a photon on an object to see how it bounces back and then understand how that object looks like, you can do the same thing with electron because electron also has both wave and particle kind of properties. And in fact, the wavelength of electrons is much closer to the objects that we are interested in than is the wavelength of visible light. So that is why electron microscopes are able to show us in greater detail the things that we're interested in, especially things like cellular organelles and deep cellular structures, right? So with that out of the way, let us look at what light microscopes are. So we just talked about light being the particle which is conveying the information about an object here, and then there are lenses used which magnify the image to finally give us the information that we're looking for. One great thing about light microscopes is that you can just put your living cells under inside your light microscope and observe them while they are carrying out their functions, while they are living and behaving like they usually do. And that being said, they have a limited resolution of less than, say, a fourth or a fifth of a micrometer. So they have a micrometer kind of a resolution. That would be like 200 nanometers, right? So while that is great, uh, that it allows us to look at things that are very small for us to see with our own eyes, it is actually not good enough to actually look at really small things like the ribosomes or individual strands of the DNA. And while I'm saying this, can you believe that there actually exist microscopes that can look at the individual strands of DNA? That is crazy, right? And another thing, so One more thing about living things is that they are transparent to light. So light will just pass through them and reach your eye, and it would be as if you're seeing nothing because everything is transparent. It's like it is passing through a glass. What structure of the glass are you seeing when light is passing through the glass? Nothing, right? And this has been this we have tried to overcome this by using dyes, which stain certain parts of the cell but not the others, so as to give it some kind of a contrast. And then we pass the light, and then we see, okay, these are the structures that exist inside uh, the cell. So the H&E stain and several others that you might have heard of are all for this purpose because unless they are applied, the cells are kind of transparent, which limits the amount of information we can get even with a light microscope. And while all the light microscopes use light as a particle, there's a difference in which the light is collected, based on which they are further classified into transmitted light microscopes and emitted light microscopes. So a transmitted light microscope is what I was describing earlier, where you have a cell and a light is passing through it, and now you're observing it. So basically, the light is originating somewhere else. It is being made to pass through the object of interest, here probably the cell, and now you're observing it after it has passed through the cell. But you might imagine that there are ways to make parts of the cell glow, and if this is being done, if you're making some part of the cell itself glow, then basically the light is coming from the cell itself, right? So it's not an external light, but it's an internal source of light, and that would be called an emitted light microscope. So here, what is generally done is that uh, uh, you have parts, you stain it with the uh, particles that would fluoresce, which means that you shine a light of one color on it, it would absorb that light and emit another color, and that would make it stand out. So while the rest of the cell might appear of the color that you're actually shining on the sample, but because this is a fluorescent material that you have stained that sample with, what it will do is it will absorb this light and emit another color of light, which will make it stand out. So these are the two broad categories into which you can divide light microscopes. And now come electron microscopes. Again, here it is just that the particle is electron, otherwise uh, the entire concept is the same. But because electrons are kind of brutal when they pass through uh, your sample, and because electrons cannot pass through the normal atmosphere, because then they would just get really sidetracked by all the atoms that are, or atoms and molecules that are uh, going around. You require to process and observe samples required for electron microscopy in a vacuum. So obviously you cannot observe living cells in a vacuum, and so basically for electron microscopy, living cells are not suitable. And while this is the limitation of electron microscopy, it offers a spectacular resolution of less than a tenth, of about a tenth of a nanometer, and that is great. The width of the DNA strand, I think, is about zero point 0.2 nanometers. So let me just double check that sizes. Yeah, so DNA is actually 2 nanometers. So we can visualize the entirety of the width of the DNA by just using uh, an electron microscope. So that is great, right? And apparently there are some uh, recent advanced microscopes that can allow you to see individual atoms. That is crazy, and probably that is not required for the sake of biological experiment, because you would be interested in all the structures, all the complex structures that these smaller atoms and molecules make up. and how they are functioning rather than individual atoms, at least for the sake of biology and genetics. And while like this then there comes the same limitation as was for the light microscope. You require some kind of labeling of the sample that you're putting for microscopy inside the electron microscope, because otherwise everything would appear the same, right? You need to make some parts of the sample stand out and some not to, so as to delineate between different parts of your sample. And that makes the sample preparation in electron microscopy very, very complicated, because you have to sometimes coat it with heavy metals, and you have to actually dehydrate the sample completely and replace it with wax or plastic and things like that. That is actually quite complicated. The sample preparation for electron microscopy, and it might take weeks to months for you to get an image after you have submitted something for electron microscopy. And like we had discussed that in light microscopy, there are two major types, namely transmitted and emitted light microscopy. In electron microscopy, there is either a transmitted or scattered. So there's no emission of electrons unless you would probably imbue a sample, a biological sample with radioactive material which emits electrons. That would be interesting. I'm not sure if it has been done yet, but the two major kinds of microscopy, electron microscopy, are transmitted electron microscopy and scattered electron microscopy. So the transmitted electron microscopy works the same as the transmitted light microscopy, whereas scattered electron microscopy works more like the way we look at objects in our room with our naked eye. So you bounce off electrons off of the sample, and then you collect those bounced off electrons and reconstruct what the object might be looking like with your computer. And yeah, so I think we'll be looking at light microscopy and electron microscopy at a bit more in a bit more detail in the next video. And this is all for today's video. Thank you.